Thanks, Arnie, and it's a real pleasure to be here. I was just talking with Ben that the first time that he and I met was actually in this room a little over 4 years ago when the temperature was about 100 degrees Fahrenheit colder than it is today. So we are straddling the extremes. And I'm going to tell you a little bit about the introduction to the work that actually Ben and Marta and our teams have begun to do together, and I think the confluence between your field and our field. A little bit of disclosure information about the companies that I've done some consulting with, and one additional disclosure that I think is relevant for this particular group, so take it easy on me, okay? But as Arnie said, we have some fundamental questions that we need to answer. And that's really the question that Lou Thomas originally posed was like, is there an immune response to cancer? And therefore, can the immune system reject cancer? I think now we know in 2019 that Lou Thomas was right, that there is an immune response to cancer, and then under certain circumstances, the immune system can reject cancer, but we still have much more to learn, and that's, I think, work that we all can learn to do together. And so the genesis of these questions really has to do with what is cancer? Is it something that more resembles a foreign infectious pathogen, something that doesn't look like the normal human body, in which case the immune system would see it as being something foreign, not self, and dangerous, react to it and reject it? Or is it self? Does it more resemble the normal tissue that it came from, and therefore the immune system being trained from the time of fetal development to ignore or tolerate normal body parts, would stand back and not respond to it? And this has really been a conundrum that our field has grappled with for decades. Like, is cancer self? Is it non-self? Or what I think is it really represents something called altered self. And those alterations in self are really what we are now studying. So a bit of a backtrack to a patient who taught us a lot about cancer immunology. So this is a patient whose two-letter code is AV. And this person presented to Memorial Sloan Kettering at the age of 17 in 1975 with a skin cancer called melanoma. So melanoma is one of several kinds of skin cancer. It's the cancer of the pigment-forming cells in the skin. It's not the most common form of skin cancer, but it is actually the most common form of skin cancer-related death. So it's the most dangerous, but not the most common type of skin cancer. If it's detected early, it's curable 80% of the time just with a simple surgical procedure. But until very recently, if it were to spread to other parts of the body through metastasis, it was almost uniformly deadly within an average of 7-month period of time. So a very lethal and virulent form of cancer up until quite recently. So certainly in 1975, there was every reason for this young man and his family to be very nervous. He had metastases to several different parts of the body. He was treated with chemotherapy, with surgery, and with experimental vaccines that were actually made from his own cancer cells, or he was injected actually with dead cancer cells from other people. Eventually, the cancer stopped coming back, and he is alive and well and lives somewhere in Colorado renting skis to people. But along the way, his cancer cells were kept alive in the laboratory through tissue culture. And one of my colleagues did a very simple experiment in the late 80s, early 90s, and combined this young man's cancer cells with his own white blood cells and showed that his own white blood cells, the patient's, could kill his cancer cells. And the question then became, as Arnie mentioned, what were the antigens, what were the targets that these white blood cells were recognizing? And through a worldwide collaboration, through the LICR, the Ludwig Institute for Cancer Research, several of these antigens were identified. And actually, this told us a lot about what the immune system could see. Actually, the first one was actually the most shocking when it was published in 1991. It was an enzyme called tyrosinase, which is a normal protein that is in all of our melanocytes, all of our normal skin cells, that catalyzes the rate-limiting step in the synthesis of melanin pigment. And so tyrosinase, we all have it. Some of us have more of it than others, but we all have it. Even people who are albino have tyrosinase, even though they make no pigment. They actually have a mutation that makes the enzyme non-functional. So the fact that this young man's T cells could recognize a normal protein found in normal skin cells and also in the cancer cells was a revelation. Okay? Second, a new protein was identified called MART1, which had not heretofore been known. This is also a normal protein found in melanocytes. And then finally, in a glimpse of what we would learn in the future, this young man's T cells recognized a mutated version of a protein called cyclin-dependent kinase 4. And so this is a potentially oncogenic protein that drives proliferation in many cancer cells, including melanoma. And this young man's melanoma had a mutation in cyclin-dependent kinase 4, and his T cells recognized an alteration in that protein. So we learned a lot from studying one patient. We learned, first, that the immune system could recognize self. Second, that a new protein was discovered. And third, that the immune system could recognize altered self. And of course, now we know the patient is free of disease for 40 plus years, which is, of course, important. So why could we not successfully immunize against these proteins? What are the obstacles to immunization? Well, unlike vaccines that target infectious diseases, antigens on cancer are largely self. Immunization with self-antigens usually doesn't work, since immune ignorance or tolerance prevents that as a way to avoid autoimmunity. So a trick that was actually theorized by a graduate student in the laboratory of one of my mentors, Alan Houghton, is something called xenogeneic immunization, or cross-species immunization. And this is really the principle, that if you're studying cancer in mice and you want to immunize against that self-antigen tyrosinase, you can inject tons of mouse tyrosinase into the mouse. And you can inject the protein, you can inject the DNA, you can inject synthetic peptides, you can use strong immune adjuvants, and nothing will happen because the antigen is self. And the mouse's immune system is not going to respond to self. However, if you begin that immune reaction with an altered version of tyrosinase, the human ortholog of tyrosinase, which is 85% identical at the amino acid level to the mouse, that difference of 15% is enough to trick the immune system into becoming activated, and the resulting T cells that have been expanded will cross-recognize the human and mouse versions of the protein and will reject melanoma in mice, and will also cause black mice to turn white because the normal melanocytes also become rejected. And so this was actually the work of a grad student, Clarissa Nafziger, in 1996. It was the subject of her PhD thesis. And so actually when I was a postdoc, I continued to study this using a similar protein called GP75, which is also called tyrosinase-related protein 1. And what we showed, this is what's called an immunoprecipitation Western blot, which looks for antibody responses, that if you immunize a mouse with mouse GP75, you get no antibody responses. But if you immunize a mouse with the human GP75, you get very nice immune responses, which are here recognizing the mouse protein. And those antibody responses are associated with a significant reduction in the number of lung metastases in mice that are injected with mouse melanoma cells. And as I mentioned before, we can cause a black mouse to actually begin to reject its own melanocytes, its own pigment-forming cells in the skin, and turn white. I think if we got the opposite to happen, if we could get gray hair to turn dark again, then we'd really have something. But we only have it going in the other direction. So this turns out to be a very potent form of therapy in mice. And as we were beginning to think about bringing this into humans, we became aware, actually, that we could apply this in pets. Because dogs also get skin cancer. They actually get melanoma, and they get it in their mouth because the haired skin of the dog actually is not a really common place to find melanoma, but the mouth actually is. And so we applied this same principle in dogs, with not like experimental dogs, but people's pet dogs that had gotten melanoma. We worked alongside veterinary oncologists. This was actually one of our first patients, this Siberian husky, who developed melanoma in the mouth and had developed lung metastases from that melanoma, shown here, pointed to by the arrows. This dog was injected with altered tyrosinase, with human tyrosinase DNA as a vaccine, and actually rejected those metastases, went on to live several years and died of an unrelated cause. We actually published this, showed actually that dogs given a vaccine after they had surgery for melanoma lived longer compared with controls. And this vaccine is actually the first licensed therapeutic cancer vaccine in the United States. It's just that it's for dogs and not for people. As it turns out, when we tested this in people, vaccines like this induce low and transient immune responses, incapable of controlling widespread metastatic cancer. And we were, of course, disappointed, but we were interested in knowing how we might augment these responses. And we began to collaborate with Jim Allison. As Arnie mentioned, Jim was a scientist at the time working at Berkeley who was studying a molecule called CTLA-4. And CTLA-4 in this diagram is this fork-like molecule. Now, when a T cell becomes activated, it actually recognizes an antigen with its T cell receptor, shown here. That signal by itself is not sufficient to completely activate the T cell. There needs to be, in addition to the T cell receptor, a co-stimulatory molecule called a co-stimulatory signal between CD28 on the T cell and a member of the co-stimulatory family of molecules called the B7 family. If you haven't learned this already, immunologists need at least 3 names for the same protein. It maximizes the chance of one person being famous. So just keep that in mind as you hear more about immunology. Once these 2 signals are delivered to the T cell, the specificity signal and the co-stimulation signal, then the T cell gets activated. But this molecule, CTLA-4, which normally sits inside the T cell, moves into this immune synapse, where it serves as a high-affinity decoy to begin to turn off the T cell and prevent it from getting completely activated. And so Jim theorized that if you could block this inhibitory molecule, you would allow for a period of more robust T cell activation. And so he developed antibodies to it. And we used these antibodies to try to potentiate the responses to these vaccines in mice. And what we showed is that you could take a suboptimal number of vaccines here, shown in blue, and significantly augment it when you gave Jim's antibody along with it. So this was a long time ago. And fast forward to last year when Jim and Tasuku Honjo actually win the Nobel Prize for their work in identifying the pathway toward cancer immunotherapy that is mechanistically represented by CTLA-4 and PD-1, two molecular breaks that normally hold the immune system into a state of homeostasis. And when these breaks are blocked, you allow the immune system to become more proliferative and more activated and to overcome some of the immune suppressing influence of cancer. So this is just another version of that kind of low-tech slide I showed you before. So just to review, the T-cell receptor signal, the co-stimulatory signal, CTLA-4 moves from the intracellular store to the immune synapse where it turns off the T-cell. If you block CTLA-4 with an antibody, you remove this break and you allow for more robust T-cell activation. One of Jim's antibodies went on to become developed as a human medicine called ipilimumab. Oh yeah, sorry, go ahead.
Is the blocker, is that a mutation or is it always there?
It translocates. It's not a mutation. Yep, it's a normal constitutively active protein that once the T cell becomes activated, it is free to translocate from an intracellular location to the surface of the T cell. Thank you. That's a great question. This is now a human medicine. It's approved for the treatment of several cancers, including metastatic melanoma. This is an example of when it works. This is a patient that I treated in 2006 when this medicine was still in clinical trials. This is a patient's leg covered with individual metastases of melanoma. There's also a melanoma as a large mass in this woman's pelvis. All of this disease regressed within about 6 weeks after 2 doses of this CTLA-4 blocking antibody. You can see that there are still some darker patches here, which we wondered whether that was remaining tumor. It's actually not. It's actually macrophages stuffed with the melanin pigment from the dead melanoma cells and no melanocytes. If we do a technique called immunohistochemistry, where we identify individual populations of immune cells using different colored staining, we can see that there are very high numbers of these CD8 positive T cells in the site where the tumor was, as well as some CD4 cells. And this is exactly what Dr. Allison would have predicted, that by blocking this molecular break, you are liberating the immune system to respond to targets that it was trying unsuccessfully to respond to with this breaking mechanism in place. And yes?
Was this patient also treated with the vaccine?
No, no. So the assumption is that these patients are being vaccinated as the tumor is growing inside them, but that immune response is not effective because of these breaking mechanisms. So the long-term survival of people with this melanoma cancer was modestly improved with the use of this CTLA-4 blocking antibody. This is data that was collected with 5 or more years of follow-up. And you can see that now, rather than having 2 or 3% of people alive for long periods of time, we now have about 20% of people alive. Still not good enough, but a step forward. As Arnie mentioned, these checkpoints, CTLA-4 and PD-1, also prevent autoimmune disease. And it was therefore not surprising that when we began to treat people with these immune modulating medicines, we began to see symptoms that resemble autoimmune disease, because we are not just cutting the molecular breaks on T cells that have specificity for cancer, but in fact all T cells that are in the repertoire. And so we may see a higher number and a higher activation state of T cells that are normally quiescent and kept in check by these mechanisms and therefore are not normally giving symptoms of autoimmune disease, but now are doing so. And so we see these symptoms in some people. We can control them if detected early with immune suppressing medicines. This is just a sense actually of all the different organ systems, and actually in a very fortunate twist for patients, the use of immune suppressing medicines given at later time points to control the side effects does not interfere with the anti-tumor activity of the medicines. And actually, on the way down here, I was actually editing a manuscript as a collaboration with some colleagues in Japan that we think that we now understand why, that the use of medicines like corticosteroids that suppress the immune system only really dampen low affinity T cells when given at late time points or early time points. High affinity T cells to some of the mutations don't seem to be sensitive to the effects of corticosteroids, which is very fortunate and very relevant to some of the work that Ben is going to tell us about. So the other pathway, in addition to CTLA-4, is PD-1. Like CTLA-4, PD-1 is a braking molecule found on T cells and actually interacts under normal circumstances with a molecule called PD-L1 on antigen-presenting cells. But tumor cells have learned that if they express the ligand for PD-1 on their surface, they actually can cause T cells that recognize them to become exhausted and to die, because this interaction between PD-L1 and PD-1 through a molecule called SHP2 silences signaling from the T cell receptor. What we can see is that PD-1 blocking antibodies can cause regression. Each of these vertical bars going down is a patient whose tumors got smaller when an antibody to PD-1 was given to them, as opposed to these patients whose tumors didn't get smaller. And in yellow are the patients who previously were treated with the other immunotherapy, ipilimumab, and didn't respond. And even though they didn't respond to one immunotherapy, they responded to the other one. So this shows us that there can be a difference in mechanism between these two immunotherapies. There may, in fact, be value in combining the two together. And this was shown in animal models of colon cancer, where combined blockade of these two molecules cured many mice. And we brought this into human clinical trials, where for the first time in people with advanced melanoma, we had more than 50% of people getting better, showing these rapid regressions with multiple metastases regressing when we used the CTLA-4 blocking drug and a PD-1 blocking drug called nivolumab. We looked at this in a large clinical trial and showed, in fact, that nearly 60% of people would get better in terms of showing regression of cancer when they were treated with the combination, higher than with PD-1 blockade or CTLA-4 blockade alone. And if we look at long-term survival, we're no longer talking about 2% or 20% of people alive, but we're talking now at 4-year time points of more than half of people alive with this otherwise nearly uniformly fatal cancer. So we've come really a long way. However, we also are challenged by the fact that when we give these two medicines together, we have nearly 60% of people with high-grade side effects. So we have to be very careful about the treatment of these people and perhaps understand who needs one therapy versus who needs two. And we haven't quite made it there yet. So now the question also becomes, why is it that some patients respond and others don't? Why is it that some types of cancer respond, and even within one type of cancer, like melanoma, why is it that 60% respond and 40% don't? And so the question that was asked is this related to genomics? And this is the bridge to Ben's part of the talk. So we know that melanoma sits at the far end of this spectrum of the number of mutations found in an individual type of cancer. Melanoma, as I said, is a kind of skin cancer, and as such, the oncogenic driver of melanoma is actually through ultraviolet light exposure in many people. And ultraviolet light is something that our skin is exposed to all the time. Most of the time it doesn't cause cancer, but sometimes it does. Nonetheless, our skin is constantly being mutated by ultraviolet light. So be careful in the summer, okay? Wear sunscreen. And melanoma has a very high number of mutations in its genome. It sits right next to a few other cancers, lung cancer and bladder cancer, which are also heavily mutated. They are heavily mutated because of chronic exposure to tobacco smoke carcinogens, which drive those cancers. And so these are also amongst the most responsive cancers to immunotherapy. And so a hypothesis was generated that perhaps the number of mutations in an individual person's cancer was related to the likelihood of response to immunotherapy. And so in collaboration with cancer geneticists at Memorial Sloan Kettering, Dr. Tim Chan and Dr. Alex Snyder, we actually looked at 2 cohorts of melanoma patients. We stratified them by whether or not they had long-term benefit from ipilimumab, in this case, the CTLA-4 blocking antibody, and we showed that in these 2 cohorts, patients whose tumors had more mutations were more likely to respond. But there were outliers. There were patients who had a very high number of mutations and didn't respond, and patients who had very few that did, implying that there are qualities of these mutations that we have yet to understand. And that has been the area of collaboration with Ben and Marta. This is also true in lung cancer when we look at PD-1 blockade. And in fact, durable clinical benefit is associated with a higher number of mutations, and in fact is associated with the presence or absence of something called the tobacco signature, which is the specific pattern of transversion mutations associated with tobacco smoke exposure. So in fact, people who get lung cancer as a result of tobacco exposure are more likely to respond to immunotherapy. But I will say this, and I cannot say it too forcefully, it's better not to smoke and not get the cancer to begin with than to smoke and get a cancer that's more likely to respond to a super expensive medicine. Okay, so, before I finish up, I'll just say that this was all retrospective data. The prospective look at this question of is there a relationship between mutations and response to checkpoint blockade was actually a very beautiful clinical trial done at Johns Hopkins, led by Young Lee, a medical oncologist, and also Luis Diaz, who is actually now my boss at Memorial Sloan Kettering, working as part of the Bert Vogelstein empire there. And they said that if this relationship between mutations and response to immunotherapy is true, then there is a small number of people who get cancer because of an inherited deficiency in the ability to repair random genetic damage that occurs every time our cells divide. So that's a frightening thought, that we're all sitting here feeling very healthy. Our cells are dividing quietly as we sit here. But as they divide, random mutations, random errors occur in the way that the DNA is replicated. Usually, that error is repaired by enzymes that are mismatch repair deficiency related enzymes. And everything goes along very nicely. However, in some people, there is an inherited deficiency in the enzymes that essentially autocorrect our DNA replication. And one of these syndromes is called Lynch Syndrome, named after Henry Lynch, who actually recently passed away. This group hypothesized that if patients with Lynch syndrome who get colon cancer and some other gastrointestinal and gynecologic cancers at a young age with high frequency, if they were treated with immunotherapy, they should respond. Whereas people with the same histologic kinds of cancer, but not due to mismatch repair deficiency, were treated, they shouldn't. And that was exactly what was observed. If you look at patients with colorectal cancer associated with Lynch syndrome and a failure of autocorrect, the number of people who have a shrinking of their cancer is 60%, and at least a control of the cancer, meaning stable disease or response, in nearly 92%. However, people with the same histologic kind of cancer, colon cancer, that does not occur because of mismatch repair deficiency, but rather because of random events, have a 0% chance of responding to the same immunotherapy. And if you look at people with Lynch syndrome, with mismatch repair deficiency, who have ovary cancer, uterus cancer, pancreas cancer, they also have a 60% likelihood of response to PD-1 blockade, implying that we should think about application of these medicines not solely based upon histologic diagnosis, but based upon the mechanism underlying the oncogenic events. And actually, several of these medicines, pembrolizumab, which blocks PD-1, and nivolumab, which blocks PD-1, and ipilimumab, which blocks CTLA-4, have now all been FDA approved for cancers that result from mismatch repair. Yeah?
Do you see a difference between somatic and germline mutations?
It's a great question. We haven't really looked at that. Yeah. Which brings us really...
What would you expect with germline mutations? In some sense, they're self, right?
Yeah. In some sense, they are self.
Yes.
So I guess the question is, in this case, have we seen immune responses to the MSH, MLH mutations, the mutations in the very proteins that are responsible for the mismatch repair deficiency? So that we really haven't seen. And I guess the other question would be looking for responses to things like BRCA, which are germline.
How does the field interpret that experiment, the experiment that showed that mismatch repair raises the frequency of response?
Mm-hmm.
You could interpret it that you'd like to have in a tumor many antigens
Mm-hmm.
and lots of T cells killing different antigens.
Mm-hmm.
You could interpret it that there's a probability of finding some particular antigen,
Mm-hmm.
or you could say that it is...
Or is it the germline allele itself that's
Or
mutant?
is it, well, you could say
Right,
that's
right. So we don't know yet. We've made this sort of hypothesis still that it's the number of mutated neoantigens that is underlying it, because that's something that's in common with the non-germline mutation-associated cancers.
In my mind, the reason that makes a lot of sense is because this whole system, the immune system, arises probably not to take care of cancer, but to take care of infectious diseases.
Yeah.
And what we know is when it does take care of infectious diseases, it sees many antigens of the infectious disease, be it the bacteria
Not
or
one.
virus. That's right.
That's right. That's right. And so this hypothesis was one that was very new the first time I came here on a cold day 4.5 years ago, right? And began a collaboration with Ben and Marta, really to understand if we could model the qualities of these hundreds or thousands of antigens, to understand what made them better or worse, so that we could interject a bit more mechanistic understanding and rigor into this. And this gave rise to some work which was a collaboration between the Institute, Memorial, and Mount Sinai, between Ben and Marta and our groups. And I'm going to hand off to Ben to go into more detail about this now. I just want to finish up thanking the many people that I work with and the folks who support us. And I want to actually thank Arnie for having the vision for bringing all of us together on that cold day many years ago, and what was truly an experiment of human social behavior to see if we all could get along and do something constructive. And I would argue that we definitely have. So, thanks.